We had just begun speaking about glucagon and insulin, these two antagonistic hormones, and then their effect on the absorptive and post-absorptive state, or as I like to call it, the fed and fasted state, okay? So we described insulin as being the hormone of the absorptive state. This was the anabolic hormone, and you want to think about a scenario of having a large starchy meal as you think about the function of insulin. Alternatively, glucagon is the hormone of the fasted or the post-absorptive state. And so you want to think about having gone for a long extended period without food as we think about glucagon's function, okay? And then we know that the sympathetic system is also heavily involved in this fed versus fasted state, right? If you're fighting or fleeing, you will not be engaging hormones like insulin, okay? You're not trying to digest food, whereas if you're in the parasympathetic state, you will be actively trying to digest food, okay? So just bear in mind that other regulators are epinephrine and the autonomic nervous system. Let's talk about the role of insulin. So insulin is a peptide hormone, first of all, and because it's a peptide hormone, this is why it has to be given as an injectable, okay? If we try to digest or orally give insulin, it will be broken down in the stomach by our relative enzymes. It's secreted from the beta cells of the islets of Langerhans. So scattered throughout the pancreas are these clusters of cells, and these are the endocrine part of the pancreas. They're called the islets of Langerhans. And among these clusters of cells, there are three types of cells. One of them are beta cells. So the centrally located cells are usually beta cells, and then the peripherally located cells are usually alpha or delta cells. And so beta cells secrete insulin, and we know that insulin has a role of storing macromolecules for energy. So again, think about that fed state. You just had a large starchy meal. So it's anabolic. It's going to sort of put some of that glucose away, use whatever the body needs at that time, and then make things like glycogen, which is the storage form in the liver and muscles, and then triglyceride, another storage form in our fat and adipose tissue. It's going to promote glucose use for energy as storage molecules, again, so putting those as sort of reserves away for the fasted state. So this increases glucose uptake into cells and decreases the catabolic state. So catabolic state is that breaking down, which is going against the action of insulin, okay? All right, so because it is a hormone of the absorptive state, it's going to increase our glucose in the plasma. It is going to increase amino acids in the plasma. It is going to be aligning with the parasympathetic or the rest and digest system, okay? It's also going to increase the release of GIP, glucose-dependent insulinotropic peptide, or glucose inhibitory peptide. And this is a protein that is released in a response to how much starch or how much glucose is in our meal. So it's released by the stomach depending on what amount of starch, how starchy the meal we had was, and so it directly stimulates insulin release to help break down some of that starch in that meal. Secretion decreases in the post-absorptive or the fed state. Again, that makes sense, right? We're not trying to store nutrients or store macromolecules away if we are fasting or if we're in the fasted state. And so the sympathetic nervous system is going to be more active here, and epinephrine is going to be facilitating that sympathetic activity. All right, so basically, if we're fasting, this is synonymous with fight or flight or freeze, okay, which is that sympathetic system. All right, so let's look at the beta cells and how they respond to increased insulin secretion. So they're going to release lots of insulin in the bloodstream in response, again, to a large starchy carbohydrate-rich meal. And the first thing we'll do is we'll mobilize most of that glucose into our body tissues, right? We're going to use what we need at the moment in tissues like the lungs or the brain, the lungs or the heart, excepting the brain, the liver, and exercising muscle, we will store those molecules away. So we're going to make some amino acids, make proteins, and then we will decrease protein breakdown or proteolysis, okay? So basically here, we are putting some of this energy away, whatever we need at that moment. In our adipose tissue, we're storing away reserves, okay? So we're creating fatty acids and triglycerides, and then we are going to decrease lipolysis, which is the breakdown of those fatty acids. Likewise, in the liver and muscle, we're storing away reserves in the form of glycogen. So we make more glycogen, increase our glycogen synthesis, and decrease our glycogen breakdown. And then in the liver, we increase our fatty acid and triglyceride synthesis and decrease our gluconeogenesis. Now, gluconeogenesis is the process of making glucose from other non-carbohydrate molecules. So this happens in the fasted state. It's sort of like a second pathway to make some glucose. And that is decreased if we just had a large starchy meal. We have enough glucose, okay? All right, so these are some of the actions of insulin. Let's talk about glucagon. So glucagon was that alternative that alternative hormone, okay, that was antagonistic in its function to insulin. It's also a peptide hormone, also released from the islets of Langerhans, but from a different cell in the islets of Langerhans. It's going to be the alpha cells, which are, again, more peripherally located in the islets, okay? Now, glucagon is going to promote glucose sparing for the nervous system. That's one of its unique jobs. It sort of harnesses or sequesters glucose and shuttles it off to the brain. You know, the brain is very unique in its glucose demand. It also mobilizes energy stores. So again, think about you being in the fasted state when you think about glucagon. You've gone for a long time without a meal, and so you're actively looking for any storage reserves for energy throughout your body. So you're going to be breaking down fats and glycogen in your muscle and liver to help mobilize some of those energy stores, so lipolysis and glycogenolysis. You're also going to be synthesizing new glucose. So again, this sort of backup pathway to make glucose from non-carbohydrate molecules. So we can use some glucagonic amino acids, and we can actually convert those to glucose. And this is what gluconeogenesis is. So we sort of tap into this secondary pathway, again, in a state of fasting if we need to find energy stores. So this is a catabolic hormone in that way. It is going to be releasing energy or releasing energy stores. Okay, so glucagon is the hormone of the postabsorptive or the fasted state. And this ties in hand with the sympathetic nervous system. Okay, so again, if you're fasting, your body actually processes this just the way it does exercise, okay? It's gonna be eliciting this fight or flight response, flooding your system with epinephrine, mobilizing those reserves, those energy stores, but it will decrease in the absorptive state. So glucagon secretion goes down immediately after you've had a heavy meal or a large meal. And that helps to increase your glucose in the plasma. Okay, so that's the sort of antagonistic role of glucagon and insulin. Insulin being the hormone of the fed state, of the absorptive state, glucagon being the hormone of the fasted state or the post-absorptive state in breaking down those energy stores. All right, so let's look at some of the functions here of glucagon. So if we increase our glucagon secretion, we increase glycogenolysis, which is breaking down glycogen. We decrease our synthesis of glycogen. We increase gluconeogenesis to make some additional glucose. We increase ketone synthesis. Ketones can also be used as a breakdown product to liberate some energy. And then we increase our protein breakdown. Again, you want to asterisk this. This is in extreme starvation mode, so we start to break down our protein. And then we decrease protein synthesis because you don't want to be, it's not cost effective or energy efficient for the body to be making proteins in a fasted state, okay? In terms of fat, we increase lipolysis, breaking down those triglycerides, breaking down those fatty acids, and then we decrease our triglyceride synthesis. Again, we're not trying to store fat in a fasted state. This is, or the principles here of the glucagon secretion is why we have the fasted diets. I don't know if you've heard of like the intermittent fasting. These are the principles of why this diet works, okay? So extended periods of fasting really shifts the body into this lipolysis state and glycogenolysis state, which is breaking down some of those excess reserves to liberate some energy. And this is also why we try to keep insulin down if you're on a diet like that, because insulin will tip that this balance off. Okay. Oops. Okay, now in terms of the tight regulation between insulin and glucagon, this is what helps to keep the blood sugar nice and stable. So normal blood glucose looks like 70 to 110 milligrams per deciliter. Glucose levels can go even higher than this, 200, 250 milligrams per deciliter, and even lower than this, 40 to 50 milligrams per deciliter. So glucose blood, and again, this is in the plasma. Plasma glucose levels can have a variety or a range, and so the function of insulin and glucagon is to narrow that range, to keep it in a very nice small window here, which is about 70 in our fasted state and closer to 110 right after we've had a large meal. So if in your fasted state, your glucose numbers are still really high, that's indicative of some type of pathology, right? Insulin is either not being released the way it should, or it's not working the way that it should if it is being released, okay? All right, and let's just look at this negative feedback loop of how glucose is regulated, how blood glucose levels are regulated. So again, if we have an increase in our plasma glucose, think about a large starchy meal, this elicits the beta cells in the pancreas to secrete more insulin. And we know one of the functions of insulin, we talked about it in that first exam material. It helps to insert some GLUT transporters into the cell. That helps with the uptake of glucose into the cells. And uptaking of glucose happens in most tissues as it's needed, right? So think about your heart cells, your lung cells. They will use the glucose as it's needed at that time. And then we can store some away in our liver and muscle for later. So we can make glycogen and we can elicit gluconeogenesis. Now, this will in turn bring our plasma glucose levels down, right? Moving that glucose into the cells. And so that will negatively feed back onto that initial stimulus of that high blood glucose, okay? And that's what makes this a negative feedback loop. Okay, so we're getting closer. Question? Mm-hmm. That's a good question, very good question. So the symptoms would be of hypoglycemia because you're not mobilizing that glucose, but you still have high blood glucose. So this is why diabetics are still fainty, are still shaky. They aren't able to use that glucose, but it's still in high levels in the blood. And that's where it becomes detrimental. It can be damaging to blood vessels, damaging to your eyes, to your fingers and toes, that excess glucose lingering around in the blood, right? It shouldn't be there. Does that answer your question? They're still hyperglycemic because we're talking about blood plasma glucose levels, but the symptoms would be of hypoglycemia, okay? Any other questions? Any other questions? That was a good question. Okay, so we were just wrapping up this slide. We talked about why this is a negative feedback because we're getting closer to our set point as we decrease our insulin. So you want to remember here that this is going to elicit an insulin response. As this goes lower and lower, as our plasma glucose goes lower and lower, this begins to elicit a glucagon response, okay, that fasting or our post-absorptive state. Okay, what we'll have left here are a few summary tables. Good study tools, again, to help synthesize some of this material. We have all of the different hormones that we talked about. We didn't speak about growth hormone too much, but insulin, glucagon, thyroid hormone, and cortisol, we did look at extensively. And these are the types of distinctions that you should be able to make with these hormones, okay?